Hello everybody, welcome back to the MDC Connects webinar series. I'm Sarah Brockbank, I'm a scientist at Medicines Discovery Catapult, and I'm hosting the session with my colleague Dan Bond. This is the second in a series of weekly webinars that we're running every Wednesday at 2 o'clock. So today, as I said, it's the second of a series of 9 informative drug discovery sessions. Each is delivered by experts at the Medicines Discovery Catapult and our CRO network partners. Each of the sessions focuses on an aspect of preclinical drug discovery, from selection of the right target, and that was the subject of last week's talk, through to identifying a hit, that's today, optimizing the compound through PK/PD, disease models, and on to safety and defining a biomarker strategy. In this second session, we're thinking about hit identification, and I'd like to welcome 3 speakers today. So firstly, we've got Trevor Askwith, who's group leader in assay biology at Domainex, who's going to provide an overview of how hit ID is approached at Domainex, the importance of quality protein, or quality compound collection, and the assay design. Then Gary Allenby, CEO at Aurelia Bioscience, is going to focus on cell-based screens, including reference to target engagement and a look to complex assays in the future. So 3D, spheroids, and Electrospun fiber-based screening are examples. And then finally, Andrew Pannifer, who's head of cheminformatics here at Medicines Discovery Catapult. And Andrew is going to describe the factors to consider in building and maintaining a quality compound collection for screening. So we will take questions at the end of each presentations. If you wish to ask a question, you can type that at any time through their talks in the Q&A box at the bottom of your screen. So not the chat box, the Q&A box. And the speakers will address these at the end of each of their respective talks. So over to you, Trevor.
Slides are good.
Slides are good. Can you still hear me?
Yep.
Yeah. So thank you, Sarah, for giving me the opportunity to talk here at this Medicines Discovery Catapult Connects webinar about the hit identification and why understanding your target is key. So just a little bit about Domainex for those who don't know. We're a contract research organisation based within the sort of southwest of Cambridge, and a biosciences hub there. We're around about 50 to 60 scientists, and we've got just over 2,000 square meters of lab space. And it's an all-in-one facility, so we've got chemistry and biology under one roof. Our clients are widely spread between big pharma, agrochemical, small, medium, and large biotech, startups, virtual companies, as well as major academic institutes. And over the years, we've developed compounds that got into clinical candidates, preclinical development. We're in cited as inventors on a number of different patents, and obviously authors on lots of different peer-reviewed papers. And we are a dedicated CRO. We've got no internal research projects, and critically, all of our staff are on one site. And that's key to this talk, is that we work incredibly closely together. As I said, we've got a comprehensive medicines research platform, so that means we've got chemistry, protein science, and assay biology all together on one site. And we cover all aspects, as you'd expect, of these divisions. So we've got computer-aided drug design, medicinal chemistry, analytical chemistry, protein expression, combinatorial domain hunting, which is particularly useful for proteins that are difficult to express, X-ray crystallography, and then within assay biology, you've got cell biology, which means we can develop bespoke phenotypic assays, biophysics, as well as biochemistry. And the teams work really, really closely together, and this is exemplified at how we go about the hit identification at Domainex. Each group isn't just siloed in its own area, so the assay biologists don't go off on their own and design a hit cascade for the target. We put the target at the centre of the project, and then we surround it by the experts from each team, so that's chemistry, protein, assay biology, all focus on the target and the target profile to put a plan in place that not only just identifies a hit that's got the best chance of acting on that drug, but they've got the best chance of turning the hit into it and translating that into a drug that's going to go into the clinic. For example, proteins that have different domains and different regions, chemistry can work out whether or not these different domains or regions are druggable, which are more druggable than the others. Do different regions infer selectivity versus other family members? And obviously this will feed into protein sciences. Do we want to produce full length protein? Do we want to produce truncated protein? And again, this will feed into the assay biology. Can we identify where the hits are binding? And do specific regions of the proteins have different downstream effects? And are we able to design a screening cascade where these different effects can be measured? And I said, all of this is focused on identifying a hit that's got the right chemical properties, not just to act as a tool drug, but to become a drug getting it towards the clinic. In terms of compound libraries, at Domainex that we've got a virtual screening platform, so that's Lead Builder, and a fragment screening platform, all of which could feed into a successful medicinal chemistry program. And successful projects need good quality starting points. To generate those, you need good quality compounds and good quality hit matter. You don't want to run screens with large numbers of artifacts or large numbers of compounds that make poor chemical starting points, because doing this means that the compounds that are good can get lost in the noise. So in terms of our virtual screening platform, obviously this can utilize structural information or homology modeling, and we tend to screen up to about 1,000 compounds in this. And at the bottom left, you can see kind of a visual representation of some of the virtual libraries we've got. And we've got a CATS library, which are all the commercially available compounds. This isn't just a virtual library that doesn't actually exist in reality. This is a library that genuinely does exist, that means that we can purchase from them, and that enables rapid hit expansion. We've also got a highly curated chemical library called the NICE library, which is a number of interesting chemical entities. As I said, this is a highly curated library. It's QC'd about once a quarter. And this is our lead-like library. So we've got in-house internal filters, and this filters out all the frequent hitters, reactive motifs, pains, and delivers Lipinski-like lead compounds. The fragment set, as you'd imagine, it's a very highly curated fragment library, diverse library, about 1,300 fragments. These are all rule of 3 compliant, and they contain a large number of 3D fragments as well as 2D fragments. We've confirmed all of their solubility experimentally at 1 millimolar, so we know that they're soluble at the concentration ranges that will be screened. And again, these are regularly QC'd and stored in nitrogen pods. And the idea of all of this is that the hits that come out of the screens are good quality compounds that are good chemical starting points. And again, it's not just about identifying a hit, but increasing the chance of actual success of the project and delivering it through into a drug. In terms of protein production, the key really is to focus on the target product profile that you want. Where do you want your compounds to bind? If you're agnostic about that, it might be better to produce full-length protein. However, it's important to produce the right and relevant protein, not just simply protein production that gives you the biggest yield. Critical to good quality protein production is a high quality QC protein. So just a single band on an agarose gel doesn't necessarily mean you've got a homogeneous protein. It's important to take it through multiple step chromatography. For example, separating phosphorylation variants by ion exchange chromatography. And again, here at Domainex, if there's ligands available, we'll regularly QC our protein by thermal shift DSF assays. Finally, in terms of the screening assay, this again, it's critically important to have good quality screening assays. And it's important to be able to assess the target in line, assess it's important that the screening assay you choose is in line with your target. So the technology that we use at Domainex regularly is microscale thermophoresis. And this is a technology that I've been appreciative of for years beforehand, before joining the organisation. And it's got a number of advantages that we feel above other biophysical methods. So it uses very, very low protein samples. So typically about 5 to 50 nanomolar concentration in about 5 microliters. This means that you can focus on the right protein. So even if your PrEP produces relatively low yield, but it's in the right conformation, this is something you can use. So you're not having to move into a preparation that just produces bulk protein if it's not the right form of protein. It's also a solution-based system, and this means that the target can be delivered in the appropriate way, not just in the way that makes the method work. It can also use multiple different labeling strategies, and often here these are directed by the structural information that we can gather about the target. And this means that we're able to deliver the protein in its near physiological state, which gives us the opportunity to be able to identify hits in a method that means it will more likely translate into a drug at the end of the project. At Domainex, we've got the supporting capabilities. So that means that we have SPR, ITC, NMR, and DSF, which obviously gives us the opportunity to put the hits through a cascade. So what I'd finally like to do is end by having a few slides just on a case study, and this is G9A, which is a lysine methyltransferase, which is involved in epigenetic regulation by covalent modification of histones. And this was a target that we wanted as an oncology target. So the only available assays for G9A were alpha screens, and these weren't suitable for high concentration fragment screening. What we also wanted to do was target the peptide binding site to gain specificity over the SAM binding domain. And what we wanted to be able to do was this we felt would be able to infer specificity over other lysine methyltransferases. What this meant was that we ended up with a complicated biophysical system because it was a 3-component system. And here with this project, we ran both virtual screen as well as fragment screen in parallel, and this is a way of screening that we find particularly beneficial. Here's the primary hit data, and this is just an example of how the hits come out of the monolith system, the MST system. And it also highlights another advantage that we find from the system, in that actually because of the way it runs, you get false positive detection occurring straight away. So on the top right-hand side, you can see an example of protein aggregation due to the compound. And then below, you've got ligand-induced photobleaching and then autofluorescence. And what this means is that when you get data out of the monolith, you can then focus on the good quality hits that come out and actually discount some of the compounds that are coming out from the noise. Going on from this project, we were able to identify high quality binding hits with really good ligand efficiency. These compounds were confirmed by NMR, STD NMR, and a number of those demonstrated positive binding. And you can see on the right that we were able to generate a number of X-ray crystal structures for these compounds. This led a successful medicinal chemistry program, improving these compounds and was keeping the ligand efficiency incredibly high. So in summary, integrated drug discovery approach, it's essential for hit ID. So having really good contacts and good communication between the chemistry teams, protein production, as well as assay biology. The most important thing is that you're not really just identifying a hit compound, but you're identifying a starting point for the drug. So the quality of the hit compound matters highly. The better the compound, the more translatable it is. It means you can more likely to have success through your lead optimization programs into delivering a drug for the drug candidate. And it relies on appropriate protein delivery, high quality compound files, as well as a sensitive screening assay that's essential for the library that you're trying to screen. And here's a final summary slide which just highlights the services that Domainex offer. So it's an integrated drug discovery service, and we've got a really good track record, not only of delivering high quality hit matter, but high quality hit matter that delivers into preclinical candidates and on into the clinic. I will now go through the Q&A sections, and I'll see whether or not we have any questions that I can answer. What are the biggest difficulties in compiling and maintaining the compound and fragment libraries? So for us, I think what we've done is the physical library enables us to, because it's not enormous, it enables us to maintain it very, very well. It means that we're able to curate it highly. I think in terms of the difficulties, it would simply be in terms of scale and having a large, the bigger the library, the more difficult it is to curate. And the first question said, what experience do all proteins work using MST as an assay platform? What percentage success rate? So Domainex specifically brought in the MST, I think about 6 years ago. So it was, we were very, very early adopters. And at the moment, I don't believe we've had any failure to develop an assay, a sensitive assay for MST. We go through quite a thorough assay dev program for our MST. So for example, we'll run a buffer screen through it, which is particularly useful. And the biggest problem with most of them is needing a good quality tool compound. And I think that's what's always the challenge, is when you have projects that come in where you don't necessarily have good quality tools or any tools whatsoever, that becomes more challenging. But so far, we've managed to develop assays and hits for, if not all, very nearly every single MST assay that's come in. So I think my time is over, and I will end these slides and hand over to Gary next.
Okay, thank you. Can people hear me?
Yeah, and we can see your slides.
Brilliant. Okay, so hello folks, thank you for joining in. I'd like to thank MDC for organising this series of interesting lectures. I'm the CEO of a small but beautifully formed CRO in Nottingham called Aurelia Bioscience. My background is mostly 30 years in preclinical drug discovery, building cell-based assays to screen compounds. So I'm going to talk about that this afternoon. Just to give you a brief outline, Aurelia Bioscience, as I say, is a small contract research organisation. We've been in business for 9 years. Myself and my colleagues started the company after we walked away from AstraZeneca in 2011. Our sort of model revolves around two aspects. On the left-hand side, we perform development of bespoke assays for fee-for-service, and those are milestone-based. On the right-hand side, we also work as FTE, so people, scientists in the lab working for clients on projects. Presentation are broken down really into 3 sections, topics and which are close to my heart at the moment. One, it revolves around Nanolook, the technology brought up by Promeda. The second one is a similar technology but for a different biology, which is target engagement, and specifically related to kinase assays. And then a third thing, which I think is really quite exciting in the drug discovery area, which is PROTAC. and I'll go through some of the projects that we've been looking at in terms of PROTAC and how we've been doing that, and then the future of cell-based assays, and more specifically looking at something we call Electrospun material. Okay, so looking at NanoBRET protein-protein interactions. So this was a published study that we did for a client. The target was something called SNORI3, which is a protein that interacts with ERK2. Specifically, the therapy here that we're looking at is osteoporosis. And what we're trying to do is we're trying to remove the brake on bone formation. So the theory is that SNORI interacts with ERK, and in doing so prevents bone formation. And so by inhibiting that, we would increase bone formation. So on the left-hand side of this slide, I'm just got two pictorials demonstrating how the NanoBRET protein-protein interaction works. So what you would do is you have two vectors. Into those vectors, you put protein A and protein B. In this case, SNORI and what it interacts with. And then in one of the vectors, you have something called nanolog. So nanolog is a luminescent enzyme that was identified by Promega and has been mutated in order to give it a high efficiency for manufacture of photons. So it's a bit like, it's a new version of Firefly. The advantage though is that it's relatively small. So nanolog itself is only at 19 kilodaltons. On the other protein, you would put a protein which becomes a halo tag when you add the fluorescent ligand. So when the fluorescent ligand is added, it binds to this halo tag protein. And then what you have effectively is you have protein A manufacturing photons because it's got the enzyme at the end, luminescent enzyme. And protein B is in a position to accept those protons, having a fluorescent ligand. So if you get BRET, which is bioresonant energy transfer, if that occurs as a consequence of those two proteins coming into close proximity, then what will happen is that you will get fluorescence. And shown in the bottom left of the slide is basically the donor signal that you get from photons around about 460 nanometers, and shown further along the wavelength at 610 nanometers is actually the fluorescence that you get from the tag. Yeah. So what you're able to do is you're able to ratio the number of photons that you're getting against the amount of fluorescence. And so that gives you a ratio which is workable from the point of view of screening. So on the right-hand side, what I've got is two graphs. The top graph, now the problem with many of these assays, as people will know, is that we don't often have tool compounds in order to play with the pharmacology, in order to make sure that the assay works. Right? And this was the case in this assay format. So what we had to do is we had to compete, we had to express and compete MEK with the SNORI and the ERK in the cells, because MEK will compete for interaction, and in that way will then decrease the signal. So what I'm trying to show here is the reproducibility of the assay. In this case, it was developed into 3841 format, and you can see that in 4 examples, 4 replicates, we get really good superimposition of those curves. On the lower graph, what you'll see is that we competed, we actually went into the cells with an inhibitor of ERK in order to try and inhibit the signal. And what you can see from there is that we get a concentration-dependent inhibition of the signal. So we went on to develop the assay into a high-throughput assay, and then to screen 50,000 compounds in this 3841 format. Top left is showing a distribution graph, frequency distribution graph of the hits, and the client picked a cutoff of 1.3%, which gave them 233 compounds. We then went on to retest those, which is shown below that, and so we got a really good retest of response 1 on the x-axis versus response 2 on the y-axis. Top right-hand side, you can see the Z factors. So in general, apart from a few blips at the beginning and partway through the screen, we ended up with Z factors that were around about 0.7, but all of them were above 0.5 and therefore acceptable. And then in the middle, you'll see that there were 3 compounds in this case that we identified that showed activity in terms of inhibiting this protein-protein interaction. What we also did was we checked out these compounds in a separate assay as in using the same technology, but not with Schnurri, which is shown on the bottom there, which is the P53MDM2. So we tested the compounds in that assay, and as you can see, there was one compound shown in orange, which actually seemed to be what I would call an assay nobler. And by that, what I mean is that the compound actually interacts with the assay biology rather than interacting with the proteins of interest. Okay, so I want to go on to a second assay, which is NanoBRET based as well, but this is a target engagement assay. So we've used this assay for looking at kinases and looking at the binding of compounds to kinases. This is a competitive competition assay between a tracer, which is a fluorescently labeled molecule that binds to a kinase of interest, but it actually occurs in living cells. Yeah, so basically you're expressing the full length kinase in the cells. That is then interacting at what you might consider physiological ATP and physiological pH, because the cell is living while the assay is occurring. And we have developed a way so that you can look at both association and dissociation rates of the compound within the cells whilst the cells are active. And obviously, by doing that, you can then look at how that activity is affected by external factors. Okay, so how does the assay work? So this is an animation here. So basically, you take your kinase of interest and put it into a plasmid, which is shown in the dark green, together with the nanolock enzyme that I had mentioned earlier on, the cDNA, and you transfect that into your cells. Leave them overnight. So in this case, we use HEK293 cells. You transfect them overnight, and then what happens is that as a consequence of that, you end up with a chimeric protein made, which basically is your kinase of interest full length linked to your nanolock enzyme. If you then add substrate for the nanolock enzyme, that substrate is converted into product, and that product is basically then light, which is then detected on a reader at 460 nanometers. If you then come in with a labeled tracer, the tracer being a compound that binds specifically to kinase with high affinity, then what happens is that you bring the tracer into close proximity with the nanolock enzyme, and the photons then activate the fluorescence, and so as a consequence of that, you'll see a signal at 610 shown in orange. If you then wash away the tracer and then come in with a competing compound, then what's going to happen is that the compound will compete off the tracer of your kinase, and by doing so, it will take the fluorescent tracer out of proximity of the nanolock enzyme and therefore the photons, and so then what will happen is that your signal will diminish. And so this is a true competitive assay, which is actually occurring within your cells. Okay, so on the right-hand side, you'll see 4 graphs. Abl kinase at the top left, FGR, EPHA8, and DDR. And what we've done here is we've competed for commercially available kinase inhibitors, dasatinib, nilotinib, fortinib, and ponatinib against these. And so what you can see is that as we compete and increase the concentration of compounds, what happens is that our BRET ratio declines as a consequence of competing off the fluorescent tracer. Yeah? And so what we can do is we can rank all of these compounds against each other for that competition within live cells. Now, importantly, what this also demonstrates is it also demonstrates that your compound can actually get into the cells. So you're looking at the permeability of the compound into the cells, as well as looking at the potency. And of course, we all know that kinase compounds are very soluble and will get into all cells without a problem. The other thing that we can do with this assay is that we can treat the cells with digitonin. And then by treating the cells with digitonin, what we actually do is we blow holes in the membranes. And then what that turns this assay into is more or less a biochemical assay. Granted, the assay occurs in the cells, but there's no barrier for the compound to get into the cells. Now, interestingly, I've said on the left-hand side there that for medicinal chemists, the success is driven by potency. So a medicinal chemist will start with micromolar and end up with low nanomolar and think that the job's all done. So the question is, what about residency time? So how long does a compound need to be bound to a target to have an effect? Yeah, and is there a differential in that residency time binding that results in some compounds having more profound side effects than others? And so we wanted to investigate that. And so the way we did that was actually to utilize a technology that we've been developing that I'll show on the next slide. So top left, what looks like a whip is actually a solution coming out of a syringe and being laid down onto a rotating drum. So this is how you manufacture Electrospun material. And this project has been done in collaboration with the Electrospinning company. Then what we can do is we can cut this material into shapes. So the shapes are shown in the black and white pictorial, the electron microscope image. So these are about 120 micron across by 50 micron thick. And what we can do is we can seed the cells onto these. Now, importantly, and I hope that this video will work, importantly, the other phenomenon about these scaffolds, as we call them, is that they are attracted to a magnet. So these are 96 well scaffolds shown in the tube. And as you can see, if I take them next to the magnet, they're dragged to the magnet and they stick to the magnet. We also have 384 well discs of material that are attracted to the magnet. So where's the advantage? So what we can do is we can seed our scaffolds with cells, right? And then what we can then do is that we can move the scaffolds around. So by moving the scaffolds around, we're effectively moving around an adherent monolayer of cells. And so what that means is that we can then transfer these from well to well. So rather than using a plate washer to go between plates and wash the cells and add the next solution, what we can do is we can actually pick up these scaffolds from wells and put them into new wells. So what we're showing on the bottom here is the ability to be able to do that by first of all soaking the cells, which are on scaffolds, in the tracer, and then taking them out whilst they're on the scaffolds, washing them in, let's say, a PBS wash, and then putting them into a well that contains, in that case, the compound, or vice versa. And then what we can then do is we can put them immediately into the reader and we can start reading. So what's shown on the x-axis in all these images, all these graphs, is time in minutes, and the y-axis is BRET ratio. And what you can see is that different compounds are competed at different rates off the receptor. Some are competed more rapidly than others. Now remember that this is a live cell assay. So what we're looking at here is competition, right, of either the tracer for the ligand, for the compound bind to the receptor, or alternatively the reverse, which is the tracer bound to the receptor, and we're trying to compete it off with the compound. Okay, so really quite interesting technology. Okay, the last thing I wanted to move on to is something called PROTAC. So PROTAC stands for proteolysis targeted chimeras, right? Now these are molecules which are shown in red there, which is called a degrader drug. Okay, and what they do is that they bind to your target of interest, right? And through a linker, they then have another binding site which binds to ubiquitin ligase, right? Now there are 3 or 4 of these that are being used at the moment. And then what happens is that because your protein of interest has been brought into close proximity with the ubiquitin ligase, your protein gets ubiquitinated, and then that ubiquitination process actually means that your protein then goes away for destruction at the proteasome shown on the right hand side. Interestingly, it also means that your compound is catalytic, because your compound doesn't get destroyed, it then goes on to find another specific target, and then it goes on to then take that to ubiquitination. So here is what the degrader molecule looks like. On the left-hand side is basically the E3 ligase binding site. You then have a linker. And then on the right-hand side, you've got your target binding small molecule, right? So that needs to bind to your protein of interest in order to then bring the complex together to form a ternary complex with the ligase, okay? And so it's really quite interesting because it means that if your protein of interest does not have a binding site, you need to find a point on the protein that will bind the small molecule, and then you could look at degradation, right? So what we use to look at that is something called WEST. So this is basically high throughput Western blotting, yeah? Rather than using a nitrocellulose membrane, what we use are glass rods, and basically shown on the right hand side is how the technology works. So you load up your cell lysate, protein of interest, protein, into your sample, into your glass rod, into what is effectively a stacking gel. You then separate it based, in this case, on molecular weight. You immobilize it with UV, and then you probe it by then aspirating your primary antibody, your secondary antibody, wash, etc. All this occurs in the instrument, and then you get a luminescent readout, right? So shown here, and I'm conscious that I'm running out of time, shown here is molecular weight on the left-hand side of what I would call a pseudo blot, right? So what we're looking at is a target that we're trying to degrade using a PROTAC. The target is shown at around about 50,000 molecular weight, and we also utilize a loading control, which is shown at about 120,000 molecular weight. So you can see the first two lanes are untreated, so our target is there and in abundance. When you add a positive control PROTAC at a particular concentration, your target completely disappears. And then shown in the next lanes are actually different PROTACs and how they've affected the target, yeah? And down at the bottom there is a demonstration that you can do a dose response in this technology and get some DC50 numbers. Lastly, coming back to this Electrospun fiber technology, one other thing that we can do with it is that we can put a fluorescent dye into the material when we're making it, which is shown in the red image. And so what that means, you can see there, is that by using something like confocal, or by using light sheet microscopy, which is shown top right, what we can do is we can distinguish the cells. Actually, you can see the nuclei there. You can distinguish the cells and the nuclei from the material. And shown in the bottom right-hand side is actually 24, 48, or 72 hours post dosing with cells in order to demonstrate that they completely colonize the material. Okay, if anyone wants more information, then please get in touch with me. My email is shown here. I'd like to thank everyone for listening, and now I'll hand over.
Right, thank you very much. Can everyone hear me?
Yep.
Excellent, great. Right, so I'm going to give an overview of applications of targeted libraries in hit identification. So targeted libraries can impact at multiple points along the pathway to clinical candidate. At the start, hit identification, then expanding the SAR around those hits straight after that. And then further down the pipeline, it will be optimizing those hits and leads against the project goals, the parameters that you've set towards those that you've set in your target product profile. Today, we're really all about hit identification, so that's what I'll be concentrating on. So with a targeted screen in hit identification, what you're really doing is trying to replace an HTS. So rather than screening your full deck of round about 1 million compounds, typically anywhere from 500,000 to 2 million compounds, you select a subset of that full deck, typically 1,000 to tens of thousands, enriched in the biological activity that you're interested in, compounds enriched with modulatory activity towards your screening target, to enable you to do a smaller, cheaper, faster screen. And to do that, you exploit your knowledge of chemical matter that's known to modulate that protein target. You can use crystal structure information, NMR structure information, cryo-EM, knowledge of the physicochemical properties of molecules that are active towards your biological target of interest. It's not advancing. Right. Okay. So the reasons for selecting targeted libraries and why you might want to do that. You might want to select a library that's particularly rich in molecules towards a particular biological target or target class. For example, kinases, serine proteases, GPCRs. So you would exploit your knowledge of molecules that you know that bind these kind of targets and your structural knowledge to enrich your library in those kind of molecules. You might be after a protein that sits in the CNS, for example, or a particular tissue or location in the body. There might be particular physicochemical properties that you will need to stick to in order to get your molecule into that particular location. Or you might be doing a mechanism-based approach. So particularly in recent years, with the arrival of these kinase inhibitors like ibrutinib and other BTK inhibitors, covalent modulators and inhibitors of proteins have enjoyed something of a resurgence in interest. So you might be looking for molecules that can covalently modify or modulate your target, or even things like transition state analogues. So all these are ways in which you can enrich your libraries in all particular properties. So how do you begin to draw together these libraries? So I think Trevor mentioned earlier, really the real essential nature of starting with good molecules. There's no point in starting off with any molecules or with many molecules that are operating through a mode of action that will not be developable. So the classic one here is frequent hitters. Now the problem with this is that at its core, with a targeted library, you are enriching your library in biological activity. And the quickest and simplest way to do that is to fill it with frequent hitters, which is not a good thing to do. You really want to avoid that. So considerable effort up front in filtering the set from which you're selecting is very important. So there are a number of ways you can do this, and the more you apply it in general, the better. So there's the well-known Payne's paper from Bale Group that was several years old now. So removing frequent hitters is something that the pharma industry has been doing sort of for a while, but this paper really highlighted it to the wider community, and is a very important concept to apply. You really don't want compounds that hit across multiple assays for really kind of bogus mechanisms, aggregation, oxidation, that kind of thing. It's important to remember, though, that the results from that initial Payne's paper came from only 6 alpha screens. So if you're using a different assay technology, you might not want to use these as kind of hard filters to remove compounds. In addition to Payne's filters, you've got other structural filters, SMARTS filters. These typically operate towards rather smaller substructural features. Lists of these have been published by the EBI and the University of Dundee. These are good ways of filtering out often covalent modulators, very reactive substances. You'll typically find lists of Michael acceptors in there. Again, important not to use these as a black box. If your targeted library is indeed a covalently modifying library, then you'll want to check that these are not being removed by the smarts filter set that you're using. So it's really worth pushing through, say, a random 50,000 compounds out of wherever PubChem or Zinc, and see which molecules these filters are trapping, as well as manual inspection of the filters. And then you've got physicochemical properties. Again, you'll want to stay within drug-like space to ensure you've got some nice start points. Also, there are plenty of undesirable mechanisms that might be particularly applicable to your target. So if you've got you're screening a target or target class, which has a catalytic cysteine, it might be particularly vulnerable to redox cycling, where molecules catalyze the production of hydrogen peroxide and inactivate the enzyme. So looking in particular for things like that would be useful. Another way, really, of looking for frequent hitters, well, the best way is looking for things that hit frequently. Look in PubChem, Chemble, to see if there's any data about compounds that you're interested in, to see if they have much in the way of biological data, or if very near analogues have biological data. So really, yeah, putting in a lot of groundwork in removing undesirable compounds before you start selecting your targeted library is very important. So not quite as bad as pains and really kind of bogus modulatory approaches like that are non-selective molecules, but you want to limit the number of these you have in your targeted library. So you might say that I want a set of molecules that efficiently addresses kinase space. That sounds like a really nice aim, something that you would want to do. But if you then sort of look at the GSK PICUS kinase assay data, which is a reasonably well populated matrix of several hundred kinases against, well, about 200 kinases rather, and about 400 molecules, I think, you'll find that you can inhibit 3/4 of those kinases with only about 4 molecules. So that's a very efficient exploration of kinase space with a small amount of chemical matter, and it's very efficient. But if you then want to have a selective molecule later on, that gives you quite a mountain to climb. So being careful on your target that you optimize on when selecting molecules is important. Here it would be beneficial to maybe restrict the number of kinases that your molecules are active towards before selecting. So thinking about how to select compounds for your targeted library, there's multiple ways. I've picked out a few here. You can do a scaffold-based approach. So this is really looking at the core scaffolds that are associated with activity towards your molecules. So if you're looking at kinases, then quinolines, pyrazoles, these are well-known active molecules. So the scaffold tree algorithm by Schopenhauer et al. Is a well-established way of identifying scaffolds in molecules. It iteratively pairs down molecules to their core scaffolds, and then you can select a level at which you're happy and use that to define your scaffold, and those which are active or associated with activity in your target class, you can do substructure searches and locate them in your full file of HTS compounds. You can then, identify them in fact any file, any collection of compound you can find. Once you've found your list of compounds containing your scaffold, you'll probably want to refine those selections. Multiple ways to do this. A very nice approach to this was taken at Biofocus. So they used, when they're building their kinase library, they refined their compound selection using crystal structure knowledge. They selected kinases in various different conformations, DFG in, DFG out, and then used this to identify points on the scaffold which they wanted to see high degree of variation, less variation. So that kind of approach is a valuable adjunct to a scaffold-based method. You can use activity modeling, so bringing machine learning into the approach. So if you have a set of molecules with known activity towards your target of interest, there's usually some biological data out there. You can query PubChem, Chembl, or your own internal data sources. You can draw together a list of compounds with either which are active or inactive towards your target of interest. You can feed those into your machine learning model. There are many of these: random forest, neural network, naive Bayes. Then apply that model to your database of molecules, and then prioritize those that show activity or predicted to have high confidence in activity towards your biological target. So in this case, I built a model using the pipeline pilot naive Bayes machine learning approach, and it has predicted these molecules to be active against checkpoint kinase. That was the particular one I chose, and they are indeed active. And this would be a way of finding compounds with high confidence of addressing your biological target. You probably wouldn't want to select simply the top N that your machine learning model predicted. You would want to look at diversity within the output and select from that with care. Now, the issue with using your existing knowledge to select molecules is that there's a danger of getting out what you put in. You'll only find the chemistry that you know about that has already been established to bind your biological target of interest, your protein of interest, and you might struggle to get interesting things. So how do we overcome that issue? Well, the scaffold and fingerprint approaches I've shown will certainly be biased towards your input chemotypes. They're very substructure driven. But there are approaches that are more dependent on the physicochemical properties and pharmacophore properties of the molecules. So this is an AstraZeneca paper a few years back now, where they demonstrated the effectiveness of using combined or fingerprints that combine these pharmacophore and physicochemical approaches, being rather less directed towards explicit substructures. Those will allow a bit more diversity in your the library that you then get. So those are libraries targeted towards particular proteins. You might also want to generate libraries targeted towards CNS. So access by small molecules to the CNS is really governed by the blood-brain barrier. And it's been shown that multi-parameter optimization approaches are very effective in allowing you to select molecules that cross the blood-brain barrier. This is an extract from a Pfizer paper that showed how multi-parameter optimization, rather than a series of hard cutoffs in your molecular properties like molecular weight, hydrogen bond donors, enables you to get a really nice compound set. So those are really some pretty typical ways in which you can select compounds for a targeted library. So the last slide really is just more of a thought provoker about how we go forward. So as I've touched on, you do run the risk of only getting back what you put in because you're looking at chemical space through the prism of what you already know. So here, for example, is a serine protease inhibitor. It's bound to its target, in this case thrombin. a very basic compound, not surprising given the strong negative electrostatic charge in the active site, but you'll see a lot of these in the PDB. You'll see more of these, also primary amines, but then all of a sudden, on the right, you see a compound, this time bound to factor 10A, and it's completely different from what we had seen before. Hydrophobic, and I struggle to see how knowledge of molecules on the left would ever enable you to predict to some pretty sophisticated molecular modeling to understand how that would bind, basically. That chlorine atom occupies a pocket which in the apo structure is occupied by a water molecule, and displacement of that water generates you quite a lot of binding energy. So really, the sort of, thought provoker is how we incorporate that really quite high level, upmarket level of computational analysis to ensure that we really recover as many of the actives from that full deck HTS for our targeted library as possible. So I think I'm probably quite close to the end. So yeah, that's pretty much what I have to say on that.
Andrew, do you want to just look at your questions in the Q&A?
Right, so Chris is silent, but no one agrees on Payne's filters. I think, yeah, that is a very good question. You'll find drugs with Payne's in there. So using them as a hard filter is certainly you certainly might not want to do that. It depends on, so if you have multiple compounds to choose from and one of them has Payne's, then, or a Payne's hit, then probably why not remove that one. If you need a compound with a Payne's filter to access a particular bit of chemical with a Payne's feature in there, to access a particularly necessary part of chemical space, then I would include it. But yeah, there certainly isn't complete agreement on what constitutes a Payne's feature. So another question, do these types of focus libraries stifle innovation? Yeah, so I kind of touched on ways in which we can try to get around just getting back what we know, getting or adding to the substructure-based and scaffold-based approaches, using more physicochemical property-directed approaches, and incorporating knowledge of the protein active site to guide your molecule selection. There will help you get around that. What is the evidence that the targeted library is any better at finding novel chemical matter than the high quality curated diversity compound set with some cost? It's all about the project aim, really. If you want to get very novel chemical matter, then yeah, you would go more towards a diversity set. If you had an idea about what you wanted, then yeah, your focus set, your targeted library would be the way to go. Depends on how many compounds you can afford to screen. If you can only afford to screen 500 compounds, then you might well be better off going for a targeted set to make sure you get a reasonable number of hits to follow up. Whereas if you could afford to screen 50,000 compounds, then a curated diversity compound set might be the way to go, depending on your project aims. So you've got to certainly build that kind of thinking into your decision right at the start.
Okay, thank you, Andrew. I'll wrap up now. Do you want to just stop screen sharing so I can just bring mine up? Right, okay, well, thank you very much to Trevor and Gary and Andrew for steering us through what was our second MDC Connects webinar in this series. Next time we're going to be looking at structural approaches to drug discovery, and there's been a few references in the talks today about that. So the first one will be proteins, X-ray crystal structures and how to get them by Derek Ogg at Peak Proteins. Then Rebecca Thompson at the Asprey Biostructure Laboratory at Leeds University is going to be telling us where cryo-EM can be positioned in medicines discovery. And then Martin Slater at Crescent is going to be taking a comp chem approach and telling us how to make the molecules that matter. So that's next week. So thank you all for attending. We've had another extremely large attendance today. Thank you all and see you next week.